Hello, my name is Owen Mandal, and I am a PhD candidate at the University of Cambridge. On behalf of my co-authors, Drs. Rafael Romero Garcia, Michael Hart, and John Suckling, I am delighted to present to you today our manuscript titled Genetic, Cellular, and Connectomic Characterization of the Brain Regions Commonly Plagued by Glioma.
In this paper, we tackled a decades-old question regarding the origins of glioma brain tumors. It has been known for quite a while that these tumors preferentially locate to particular regions of the brain, but it is unknown why this is, despite the clear relevance of the question for the clinical management of these types of tumors. In this study, we mapped the brain regions commonly frequented by gliomas, then quantitatively tested 3 hypotheses to probe the connectomic, cellular, and genetic etiologies of the disease. First, we constructed a quantitative map of glioma distribution by warping 335 tumor masks from a publicly accessible database onto a common template. This map demonstrated that gliomas are non-randomly distributed, appearing most frequently in dissociation cortices, specifically in the temporal, insular, and frontal regions of the brain. Next, we tested the idea that the tumor distribution is related to hub connectivity. The brain is a richly interconnected network, which can be described in terms of anatomical connections, as shown here, as well as functional connections. It has been shown that the brain regions which form many important connections, which are called hubs, are vulnerable to a variety of pathological processes due to the metabolic costliness of maintaining high connectivity. By estimating patterns of functional connectivity, which is different from the structural connectivity shown above, we constructed a network graph conveying connections between each pair of brain regions. By counting the number of connections each region in the graph makes, we can separate the regions into hubs or non-hubs based on whether their connectivity is higher than average. If we plot hubness onto the brain, and instead let the variable vary on a spectrum as opposed to being binary, we get a map that looks like this. We demonstrated that this metric of hubness, along with a number of more sophisticated metrics, correlated significantly with our tumor frequency map, providing evidence for the idea that brain network hubs are vulnerable to oncogenesis. Then, we tested the idea that gliomas are more frequent in brain regions enriched with their putative cells of origin, neural stem cells and oligodendrocyte precursor cells. Neural stem cells in the adult brain are rare, but can be localized to neurogenic niches in the dentate gyrus of the hippocampus and the subventricular zone. We identified the tumor frequency in brain regions adjacent to these niches, the hippocampus and the caudate, and found that gliomas were significantly more common in these regions compared to randomly selected pairs of brain areas. We also compared our tumor distribution with a map of brain regions estimated to be enriched with oligodendrocyte precursor cells, and again found a significant correspondence. Next, we leveraged an atlas of postmortem gene expression across the human brain to identify genes with spatial transcription patterns that matched our tumor distribution. We found 2 distinct components of gene expression which correlated with glioma frequency in the subcortex and cortex respectively. The first partial least squares component was enriched for genes involved in chromatin organization, while the second was enriched for genes involved in metabolism and synaptic signaling. Interestingly, the first PLS component was also enriched for a set of proto-oncogenes known to cause glioma tumors when mutated. Finally, we were interested in how each of these factors combine to determine glioma distribution. We combined the connectomic, cellular, and genetic factors into a regression model and found that it explained 58% of the variance in glioma frequency. It is important to note that the amount of variance explained by the PLS components is inflated by construction, but nevertheless, these results imply that each factor contributes significantly in describing the brain regions vulnerable to oncogenesis.
In conclusion, we have demonstrated that glioma frequency can in part be explained by functional hubness, distribution of stem-like cells, and transcription patterns of genetic drivers of gliomagenesis. These results add to previous literature reporting the vulnerability of hub regions to disease and provide support for stem cell theories of glioma. Our findings highlight the importance of bridging diverse scales of biological organization, from genetic to connectomic, in the study of neurological dysfunction.